And now it's time to launch the presentations and step into the topics of the day, and this morning especially about tools development with Fabrice Vavre from Lyon 1 University. Fabrice Vavre, you are a CNRS researcher, director of the Laboratory of Biometry and Evolutionary Biology and co-coordinator of the Equipex Plus project InfectioTron you're going to introduce. Your research deals with the ecological and evolutionary dynamics of host symbiont interactions in arthropods and the potential use of symbionts to control pests or vectors of importance in plant, animal, and human health. So now, let's tell us about InfectioTron, integrative tools for the study of infectious diseases from the wild to the lab. It's up to you.
Thank you very much, Fabrice, for the kind introduction, and good morning to everyone. First of all, thank you very much to the organizers for this great conference and this invitation to present InfectioTron. So this project that was indeed recently funded by the ANR under a French call that is called Equipex Plus. And it's very interesting for me to present this project in this session regarding the tools that are required to develop the One Health approach. And this is also because we have heard great talks and great initiatives yesterday on how to implement at a very global scale the One Health approach. And what I will propose here is much more modest, but is also maybe more pragmatic on what could be developed locally to go beyond data and to allow the study of specific systems and to accumulate knowledge and increase preparedness on specific systems. So just to be clear, InfectioTron does not yet cover the entire field of One Health, but at least implement at its level the first principle that we want to settle here in Lyon at the academic level, and that hopefully will serve for future development to complement it. Well, okay. I won't take long to define the One Health approach, as this was already largely done yesterday. So the link between human, animal, environmental health is now evident. And of course, there are two of the clearest examples of this, which are the reemergence of infectious diseases and the evolution of antimicrobial resistance. Tackling these questions require both a clear understanding of what is going on in the field, but also to understand the more intimate mechanisms of the biological interactions that are at play in these systems. And this is really our objective in InfectioTron, how to link nature to the laboratory and to define clearly the mechanisms that are at play in these interactions and problematics. So in order to do that, we know that we need some ingredients. The first ingredient is an interdisciplinary community and how to put them together. And to put them together, we also need complementary and integrative tools and equipment. And the last ingredient required is a favorable ecosystem in which to implement this research. And we are fortunate in Lyon to have this strong interdisciplinary community. As François Loïc showed yesterday, so 10 years ago, we've developed within the frame of the Labex EcoEffect this community, which involves different competencies on different campuses here in Lyon, with the first one on the Lyon Tech Ladois campus, where we develop ecological and evolutionary approaches that include population, community, and ecosystem levels, and that deals with the study of vectors and environmental reservoirs, as well as plant health. Interestingly, we also have forces here regarding methodological developments in statistics, mathematics, and informatics, which is, of course, one of the main strategic problem that we may face regarding data collection and analysis. The second approach is the veterinary approach that is developed in Marcy on the VetAgro subcampus, with, of course, a strong involvement in animal health, through the development of epidemiological and physiopathological approaches, and of course, the study of animal reservoirs. And finally, on the Gerland biodistrict, the approaches include the study of cellular and molecular interactions of host microbiota pathogen interactions. It also includes immunology, and I would also add the importance of clinical approaches in relation to hospitals, and therefore a strong connection with human health. One thing that might be missing, that is missing, are humanities. This is something we are working on it at the moment, but that will complement these different approaches that we are developing here on the site. Okay, so this is the competencies that we gather on the site, and now the other thing that we want to develop is a set of complementary and integrative tools and equipment, so that these different competencies can work together. And this is indeed the ambition and the main goal of this InfectioTron project, which is to ensure within the same infrastructure a multi-scale and multimodal analysis of infectious events from their natural environment to the analysis of the cellular and molecular mechanisms of infections in animal, human, and plants, with the idea to develop a coordinated network of innovative equipments and complementary platforms integrated into the same ecosystem. And of course, the idea is to set up new tools, new equipments in high security confined facilities, BSL3, A3, I3, to be able to work on specific and important pathogens for human and animal health. So the project is occurring for the next 8 years. It's a little bit more than 4 million euros. It gathers 10 different research units, 4 platform federations. So we co-lead this project with François-Luc Cosset, Fabienne Archer, and Estelle Loukiadis. So the project is coordinated by the University Lyon, but also gathers quite a lot of different organisms and university, ENS Lyon, Bête AgroSup, of course, INSA, INRAE, CNRS, INSERM, and the École pratique des hautes études, which shows you the different disciplines that we try to gather in this project. And I won't take long to describe all the different equipment that we want to buy with this project. But the main idea is really that on each site, we want to develop specific tools that are really important for developing this One Health approach. So, for example, for the veterinary approach, what we want to do is develop facilities for new animal models, and notably a bat rearing facility. We also want to equip a kind of very big population cage of different rodent species that live in almost wild conditions, and to equip this with different monitoring systems to follow these populations. But we also want to finalize the equipment of an autopsy room in BSL3 conditions, so that we can also work on wild fauna and on epidemiology and physiopathology on wild fauna. Regarding the Ladois campus, the different equipments are dedicated to develop automated high-throughput molecular diagnostic of infectious samples, so on different matrices like tissues, blood, plants, arthropods, etc. And also to develop live imaging on arthropods with a kind of confocal microscope. And finally, on the Gerland biodistrict, the idea is to develop new nodobiotic boxes equipment to study microbiota and the interaction between microbiota, wild fauna, and pathogens. So this is quite important to work now on more natural systems than lab mice and their specific microbiota. The idea is also to develop in vivo and in total live imaging systems in BSL-3 conditions to identify pathologic factors, but also to develop automated analysis of living cells to perform time-lapse study of viral and bacterial infections. And finally, one of the big equipment is a cryo-electron microscope to study both structural questions about host pathogen interactions, but also more cellular interactions. So all these equipment may allow InfectioTron to provide all the systems for molecular and anatomic diagnostics of infections in a variety of organisms like vertebrates, arthropods, plants, through an increase of our capacity to discover, detect, and diagnose at high throughput circulating pathogens, phenotyping of BSL-3 pathogens, and of course the study of transmission, host responses, and disease progression in experimentally and naturally infected small animals vectors, in toto, but also as well as in living tissues, cells, and in pathogen-contaminated biological fluids. So all this system is very integrated and is also integrated in a very favorable ecosystem because beyond the academic partners, we also are here in Lyon within a very fantastic ecosystem. So I won't present it in detail. There are too much things in this slide, but the important thing is that we gather different academic partners, surveillance services, hospitals, of course, the hub, and that is present here, and of course, important industry partners that makes the site an ideal place to develop this kind of projects. The civil society is not in this slide, but is of course particularly important to involve also in this project, and this is something that we should work on more specifically. So all in all, we hope to progress in the right direction by proposing this integrated ecosystem to work on the One Health approach in a very pragmatic way. And we hope that these ingredients will allow to tackle different pragmatic objectives for predictions, for surveillance, for prevention, but also for therapeutic developments. To give you just maybe a flavor of what we can do in this system, I wanted to give you this example. So we are currently developing with different partners project on autonomicviruses. You may know that they have been identified as a major threat by WHO. And we have assembled a consortium that allows us to study these viruses from their vectors and reservoirs to the study of their interaction mechanisms with the organisms and cells of different host species. And we hope that by putting these different competencies together with the equipment that we have now settled with InfectioTron, we will be able to accumulate knowledge on these specific systems, and that this will allow to be better prepared in case of the emergence of this kind of pathogens. And this is very similar to what Teshome discussed yesterday about how working on specific systems may really help to be better prepared in case of a new crisis. And with this, I think I will finish, and I thank you for your attention.
Thank you very much, Fabrice Vavre, for this presentation. I have a few questions for you, and I remind everyone connected right now on this conference that you can use the question window, which is just on your bottom right, normally on the bottom right of your screen, and I see that two people already have asked a few questions. So the first question would be the integration of tools and skills for studying zoonoses can play an important role to increase our preparedness against emerging menaces. Could you propose a practical example of how the InfectioTron initiative might help understand or even detect a new menace faster?
Okay, so this was the idea of this last slide on autonomicviruses, the idea is really to put together different competencies. So, for example, in this project, we gather people that work on wild fauna and the following of wild fauna, for example, boars and this kind of animals, but also that study how, for example, the evolution of oaks production is something that is very important, for example, to give different resources to different wild animals, and how this could also play a role in the population dynamics of these animals and may facilitate the propagation of different pathogens. And the idea is to tackle this question by following these different populations in the field. So take samples on wild fauna, arthropods, check them and use the diagnostic tools, that we are proposing in InfectioTron to check for the presence of viruses in these different animals. But it's also to be able to develop model systems in the lab to study the interaction at the molecular level between these kind of viruses and different species and host species, and to integrate that in the same proposal and within the same framework. So I think that this is something that is important, that the different competencies can work on a single system from very different angles. And to put that together is something that we propose in InfectioTron, and that will be possible with this tool.
Okay. I'll get 2 or 3 other questions. Maybe you could be a bit short so as to get enough time to ask you all the questions, because we're very short of time. And for example, Cédric Desier says, thanks, Dr. Vavre. And as for bat rearing facilities, how many different bat species are you going to raise? And do you intend to have bat reproduction or just picking up animals from the wider environment?
Okay, so this is one of the big questions. So we are not yet clear on the species that will be reared here. So at the moment, the facility is not settled yet. So we still have some time to think about that. The idea would be to focus on few species, very few species, and to go to reproduction if possible. But this is a very this is a big challenge, but that's the idea.
Okay. Xavier Bailey asks, to which extent resources of InfectioTron and private partners are complementary?
Well, that's also a very good question. Thank you, Xavier. Well, I guess that we have now different tools to better integrate all the tools between the academic and private partners. Well, I guess that maybe there are some resources that are very specific. I think about the cryo-electron microscope. That could be something quite interesting also for private partners, maybe, especially because it will be positioned in a confined environment, which is quite original. So this might be something that is quite specific for InfectioTron. But I guess that you are right in the sense that we have worked at the academic level to organize things. We have to probably also do that also with an industrial partners.
Very briefly, when would the cryo-EM be available?
Good question. So, well, we hope that it will be available in 2024 because we need to build a new building to host it, and so hopefully 2024.
And this question education is also a major issue in the development of One Health approaches. Do you expect InfectioTron to result in new education programs for physicians, vets, biologists, and has this been already planned in the project?
Okay, this is also a very interesting question. So indeed, InfectioTron is one piece of the global strategy on the development of One Health here in Lyon, and we are thinking about indeed the global picture. So once again, we had the Labex EcoEffect, which was at the scientific and research level. There is InfectioTron that is more dedicated to the equipment, but indeed there are reflections to work on specific training on One Health, both in Lyon, but also in VetAgroSup. And there are already some new parcours that are developed on the One Health approach.
Okay, thank you very much, Fabrice. And anyway, you stay with us because you will be participating to the roundtable at about 10 o'clock with other speakers. So, of course, you will be able to continue talking and answering questions. Now it's time to welcome Rosangela Tozzoli. Rosangela, you are a molecular microbiologist, researcher at the Italian National Institute of Health, Instituto Superiore di Sanita. You're a member of the staff of the European Union Reference Laboratory, a national reference laboratory for Escherichia coli, and you're going to talk about that and Shiga Toxin producing Escherichia coli in food. It's up to you.
Good morning, everybody, and I'm very glad to be here at this conference, and thanks to the organizers for inviting me to share the experiences that we have had at our laboratory, acting as reference lab in the development of methods. And I thought that this would be a good point to discuss because this session is actually about the tools, and so I thought that that sharing this experience would be useful. So the European reference laboratories have been nominated in the area of food safety and animal health with the regulation on food controls that was first issued in 2004. This regulation has undergone several revisions, and in this last one we have detailed characteristics and duties of the European Union reference laboratories, and which I have reported in this slide. And as you can see, the duties are quite a few, but I would like to underline that one is very, very much pivotal, which is the production, the development and the production and the provision to the national reference laboratories with the analytical reference methods. And this is really a key activity of our laboratory. Of course, among the activities, there are also the support of national reference laboratories operating in the areas of interest, and there is also the collaboration with some agencies such as European Food Safety Authority, European Center for Disease Prevention and Control, and of course with the European Commission, providing them technical and scientific assistance. Actually, in each member state, there are national reference laboratories which mirror quite a bit the duties that are of the European reference laboratories, because they are collaborating with the URLs, but they are also providing the analytical methods that they receive from the URL to the official laboratories operating in their countries. And so by the constitution of this network of reference laboratories, we have the transfer of knowledge from the European reference lab working at EU level to each member state and to the local level, so ensuring that the harmonization of the use of the procedures and also ensuring the networking and the collaboration. So as for our laboratory, as I was saying, we are dealing with pathogenic E. coli and specifically with Shiga Toxin producing E. coli, which are severe foodborne pathogens because they are of high public health concern because they can produce infections with severe symptoms, and they can be also fatal sometimes. And they have been linked either to sporadic cases or large community outbreaks. And they have this zoonotic origin, and cattle has been recognized as the main reservoir. So as anticipated earlier, the URL for Ecoli is located and hosted at the Instituto Superiore di Sanità, and it was appointed in 2006. And I started to work since that day in this laboratory, and it also operates as the Italian National Reference Laboratory. I would like to share here in this presentation the problematics that you face when you develop a method. So the first thing you have to do is to define the targets. And then once you have selected the targets, you want to define the reagents, the conditions that you want to include in the method that you are developing. And then you have this pivotal point, that is the determination of the performance parameters of the method you developed. And I would like to underline how much we have the support of the national reference laboratories in all these, in the methods development, and particularly for the determination of the performance parameters. And I will show you in a while, but first I would like to say a few words about the standard method that has been developed for the detection of Shiga Toxin producing E. coli in foodstuff. It was published in 2012, but the project started in 2006 when the URL for E. coli was appointed to chair this project. And it has the draft of this standard has been used since 2009 in the interlaboratory studies carried out by our laboratory, and it is still applied during our scheme of interlaboratory studies. And based on the results that we obtain each time we perform a proficiency test, we have included these results in a document reporting the performance parameters of the standard, as it has not been validated yet. Another thing that I wanted to share with you is the fact that in 2011, I think that you probably all know that there has been a very big outbreak caused by an E. coli, Shiga Toxin producing E. coli, which was linked to sprouts. And so in 2013, the European Commission has issued a regulation which actually set the microbiological criterion for STEC in sprouts. And in this regulation, for the first time, there was the possibility to test spent irrigation water for the detection of STEC. So the food business operators could either test the seeds, the sprouts, but also the spent irrigation water. And this was quite a challenge for us because there was not a procedure that was for the pretreatment of these metrics that was available. So at the European Reference Lab, we had to deploy this. And this is quite a tricky metrics because it is colloidal. It is quite difficult to think about making a filtering for these metrics, and so we had to think about and make some tests about different pretreatment procedures, and we decided to propose actually centrifugation. And before, and then the enrichment, and before putting this treatment in the workflow of the standard that has been already issued. And how could we test the effectiveness of this procedure? By evaluating its performance parameters during interlaboratory studies that we proposed to our network of national reference labs and also to the Italian official laboratories, which have been very much collaborative with us in understanding if the procedure was fit for purpose. So the first proficiency test that we carried out was number 16. It was carried out in 2013, and it showed performance parameters that let us think that this pretreatment procedure could be quite effective. But later, with another proficiency test that we carried out, we understood that we needed some adjustments, and we made a modification also for the for other parameters, such as the enrichment temperature, and the modification included seemed to be effective for the fit for purpose of the method. So actually, this is just an example to show you how we can refine a procedure thanks to the collaboration of the network. And now we have this standard that I told you that has been actually issued in 2012 and is currently under revision. The revision is carried out by a group of experts that has been established at SEN level. Nonetheless, we also have requested some support to the National Reference Laboratories Network by administering some surveys, by including the modification, by including some modifications of the method in interlaboratory studies to see the performance. And so this was really very, very important to have the collaboration of these laboratories, which is really crucial for the methods development and revision. I would like now to carry out, to go on with the new technologies that are available. We have been talking about whole genome sequencing, so the technology advances, and we changed the syntax of the results that we have been working with during the years. So we were talking about PCR, or we have been talking about the evolution in real-time PCR, and then we had the chromatograms and the sequencing, but now we are talking about very, very high throughput technology like the NGS, which produces a lot of data, but which produces the FASTQ files, which are quite difficult to understand if you don't use specific tools. And so in this respect, we had deployed a Galaxy-based public server termed Arise that is available. It's open source. It is a public server which has quite a user-friendly web interface, and that has been developed by experts of our laboratory, and which contains very, very good tools, important tools for the data mining, for NGS data mining and analysis, particularly for Shiga toxin producing E. coli and pathogenic E. coli, but not only, because this system, of course, is customized, and also other tools for other microorganisms can be uploaded and used in that. So the ARIAS geographic spread, this tool is the login to this web platform is not restricted to national reference laboratories, but we also have requests from other laboratories that we, of course, have been accepted. And as you can see here, it is distributed, it is used worldwide. We are quite happy for this, and we hope that in the next future, also the use of the platform will increase as well. And so just a quick look. So it appears like a normal Galaxy web platform, and it has several data mining tools, such as assembly, quality check, mapping, and things like this, but also tools for E. coli typing. And I would like to mention that we have developed also pipelines, which are very useful tools for analyzing from the FASTQ files directly to have the characterization of the strain that you are studying. So I didn't mention them here, but they are available in 2 areas. And so just a few concluding remarks. I don't want to still time to the discussion. So I just wanted to share with you our experience in the tools development. And as said, the determination of the fit for purpose of the methods proposed is really a key issue. And we have seen in our laboratory how much the network's collaboration has been useful to determine the performance parameters and also in the revision process of methods. And we are always trying to provide new methods to the network, which may face new challenges, and particularly also in the analysis of the new technologies outputs, which are currently used more and more from each laboratory. And I also would like to conclude that the scientific support and the analytical support can be, of course, shared in the network. And so this is also a very important key issue. So I finished with that, and I would like to thank you for your attention.
Thank you very much for your presentation, Rosangela. It was very interesting, as usual, and anyway, every time we learn so many things, so I'm sure that the roundtable, just a few minutes, will be very rich and very exciting. As we are out of time and already late, 15 minutes late, I would just ask you if you agree one question. Your presentation showed how important is the collaboration network around a central reference laboratory. How did network emerge initially? Did it result from a voluntary initiative from a central EU institution?
Actually, every national reference laboratory has been assigned from the competent authority of each member state. So in this sense, we have been actually informed about the nomination of these laboratories. And so it was very nice because the first year, what the main activity that we did was to start knowing the network. So we just had a workshop and we started to have communication. But I have to really say that during the years, this collaboration has been always and always reinforced. And I also would like to mention that it is very, very much important that the communication of the procedures and of the state-of-the-art methods that are distributed is also actually distributed at the national levels. And so we really thank also very much the collaboration of the NRLs in this sense to make the spread of the knowledge work very good.
Thank you.